Welcome back, future doctors and biology enthusiasts. Get ready, because today we're taking an anime-style cinematic journey straight into the control center of the cell. Yep, in this explainer, we're diving deep into Nucleus Part 2. We're going to unravel the ultimate mysteries of how the cell manages its absolute most precious cargo. So grab those notebooks, get focused, and let's jump right in.
Now, I really want you to think about this for a second. DNA को इतना लंबा thread cell के अंदर कैसे fit होता है? Seriously, how does such an insanely long thread of DNA fit inside a microscopic cell without turning into a totally tangled nightmare? It's the ultimate space-saving biological mystery. We're dealing with microscopic dimensions here, but the sheer volume of information stored inside is literally mind-boggling.
Okay, let's look at this number, because it usually shocks people. 2 meters. A single human cell contains approximately 2 meters of DNA thread. That's probably taller than a lot of you watching this explainer right now. And the cell has to distribute all of that massive 2-meter thread among just 46 chromosomes, or 23 pairs. Think about trying to pack 2 meters of thin fishing line into a tiny, invisible microscopic bead. No way, right? How does the cell pull off this architectural miracle? Well, to figure that out, we first need to meet the players inside the nucleus.
Section 1. Nucleolus, the ribosome factory.
So before we talk about the DNA packaging itself, we've got to talk about what else is floating around in the nuclear matrix, which is also known as the nucleoplasm. The first major player is the nucleolus. Think of this as a heavy-duty biological manufacturing plant sitting right inside the nucleus. But it's not quite like the other organelles you've studied. Massive NEET alert right here, guys. Burn this fact into your memory. The content of the nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane-bound structure. So many students lose easy marks here because they just assume everything inside the cell has a protective bubble around it. Not the nucleolus, no membrane at all. It's just a dense, spherical region directly mixing with the nucleoplasm around it.
So the crucial question is, what exactly is this non-membrane-bound factory making? Well, it is the site for active ribosomal RNA synthesis, or rRNA. If you look at cells that are actively carrying out a ton of protein synthesis, the bodybuilders of the cellular world, you'll find that their nucleoli are much larger and way more numerous. More protein demand means you need more ribosomes, which means you need a bigger, highly active nucleolus factory to just churn out that rRNA.
Section 2. Chromatin and Histone Proteins.
When a cell is in its interphase, meaning it's just living its everyday life and not actively dividing, its genetic material isn't neatly packed up. Instead, the interphase nucleus contains this loose, indistinct network of nucleoprotein fibers called chromatin. Let's look at the ingredients of this chromatin. It's a mix of DNA, RNA, some non-histone proteins, and absolutely crucially, basic proteins called histones. Remember our mystery of the 2-meter thread? Well, histones are the secret weapon. The DNA wraps around these basic histone proteins to form structures called nucleosomes. It literally looks like beads on a string. If DNA is the thread, histones are the spools. बिना histones के nucleus के अंदर tangled earphones जैसा हाल हो जाएगा. This thread wrapped on spools keeps everything perfectly organized. And a quick fun fact for your exams, this chromatin isn't uniform. The loosely packed, transcriptionally active regions are called euchromatin, while the densely packed, inactive regions are heterochromatin.
Section 3. Anatomy of a chromosome.
But when it's finally time for the cell to divide, that loose network of chromatin just won't work. It's way too messy to split evenly. So during the different stages of cell division, the chromatin condenses. That thread wrapped on spools gets packed, folded, and supercoiled until it forms the highly structured, easily movable packages we call chromosomes. Every single chromosome essentially has a primary constriction, like a pinched-in waistline, called the centromere. This centromere holds the 2 chromatids of a chromosome together. But notice the disc-shaped structures on the sides of the centromere. These are called kinetochores. They are essentially the grappling hooks that the cell uses to pull the chromosomes apart later on during division.
Now, while every chromosome has a primary constriction, sometimes a few chromosomes have an extra pinched area called a secondary constriction. These are non-staining and appear at constant locations. This extra pinch gives the very tip of the chromosome the appearance of a small, separate fragment, and we call that little fragment a satellite.
Section 4. The 4 chromosome types.
We know chromosomes have a centromere, but here's where it gets really fun for exams. We can classify every single chromosome into one of 4 specific types. And the classification is based entirely on one simple thing, where exactly that centromere is parked. Let's break them down. I absolutely love using memory tricks for this. First up, we have the metacentric chromosome. Meta means middle. The centromere is right in the middle, giving 2 perfectly equal arms. Next is submetacentric. The centromere moves slightly away from the middle, creating 1 shorter arm and 1 longer arm. Then we have acrocentric. Think almost at the end. The centromere is situated close to its end, forming 1 extremely short arm and 1 very long arm. Finally, telocentric. Think terminal or tail. A telocentric chromosome has a terminal centromere right at the very tip.
Let's lock that in mentally right now.
If you picture the evolution from perfectly balanced to completely one-sided, it makes total sense. First, we have the perfectly balanced metacentric chromosome. Then, as the centromere slips up slightly, that's your submetacentric. Keep going, and you get the acrocentric with its tiny, almost stubby short arms. And finally, the extreme end is the telocentric chromosome, where the centromere is totally terminal. See how perfectly the memory trick matches up?
Section 5. NEET Target. Rapid Fire Quiz. All right, it's time to test your active recall. Let's go.
Question 1. What basic proteins are found in chromatin alongside DNA? I'll give you a second. Remember, the spools for the thread? You got it, histones. Those basic proteins are absolutely crucial for packing that 2 meters of DNA.
Question 2. True or false? The nucleolus is a membrane-bound structure. Think back to that flashing NEET alert earlier. The answer is false. Its content is continuous with the rest of the nucleoplasm. It is simply a dense factory for rRNA synthesis.
Question 3. Which chromosome type has a completely terminal centromere? Remember the memory trick for terminal or tail? Boom! Telocentric. Exactly right.
Okay, final question. Let's bring it home. What is the small fragment appearance caused by a secondary constriction called? It's sitting right at the tip. It's the satellite. Fantastic job if you nailed those.
You guys absolutely crushed it today. We unraveled the mystery of how 2 meters of DNA fits into a cell using histones as spools, we toured the Nucleolus ribosome factory, and we mapped out the exact anatomy and types of chromosomes. Now, keeping all this fresh is key. In our next lecture, get ready, we are going to do a complete chapter revision in one shot. It's going to be epic. But before you go, ask yourself this. If just one tiny secondary constriction can create a satellite, what happens to the entire human body if a kinetochore grappling hook completely fails to attach during cell division? Think about it. Until then, keep reviewing, stay curious, and keep studying smart. See you in the next explainer.